Hello everyone, and topic for today's discussion is beta oxidation of fatty acid. Beta oxidation of fatty acid is the breakdown of a fatty acid to acetyl-CoA. It occurs in the mitochondria, and this process is strictly aerobic. After production of acetyl-CoA is fed directly into the Krebs. It occurs in many tissues including liver, kidney and heart. Fatty acid oxidation does not occur in the brain. It has three types of fatty acid oxidation: beta oxidation of fatty acid, alpha oxidation of fatty acid and omega oxidation of fatty acid. The beta oxidation of fatty acid involves three stages. First is activation of fatty acids in the cytosol, transport of activated fatty acids into mitochondria, also known as carnitine shuttle, and third step is beta oxidation proper in the mitochondrial matrix. In the first step, activation of fatty acid, and this proceeds by fatty acid thiokinase, also known as acyl-CoA synthetase, and it's present in cytosol. For this thiokinase, it requires ATP, CoA-SH, Mg2+. And the product of this reaction is fatty acyl-CoA and water molecule. Second step is the transport of fatty acyl-CoA from cytosol into mitochondria. It is a long chain. Acyl-CoA traverses the inner mitochondrial membrane with a special transport mechanism called carnitine shuttle. Acyl group from acyl-CoA is transferred to carnitine to form acyl-carnitine, catalyzed by enzyme called carnitine acyltransferase 1 in the outer mitochondrial membrane. Acyl-carnitine is then shuttled across the inner mitochondrial membrane by a translocase enzyme. The acyl group is transferred back to CoA in matrix by carnitine acyltransferase 2. Finally, carnitine is returned to the cytosolic site by translocase in exchange for an incoming acyl-carnitine. Third step is the proper of beta oxidation in the mitochondrial matrix. It happens in 4 steps. Step 1, oxidation by FAD-linked dehydrogenase. Step 2, hydration by hydratase. Step 3, oxidation by NAD-linked dehydrogenase. Step 4, thiolytic cleavages by an enzyme thiolase. The first reaction is the oxidation of acyl-CoA by an acyl-CoA dehydrogenase in order to give alpha, beta unsaturated acyl-CoA, also known as enoyl-CoA. It is an FAD, is the hydrogen acceptor. The second reaction is the hydration of the double bond to beta hydroxy acyl-CoA, also known as para-hydroxy acyl-CoA. The third reaction is the oxidation of beta hydroxyl-CoA to produce beta keto acyl-CoA. It's an NAD-dependent reaction. The fourth reaction is the cleavage of two carbon fragments by splitting the bond between alpha and beta carbons by an enzyme called thiolase. The release of acetyl-CoA leaves an acyl-CoA molecule shortened by two carbons. This acyl-CoA molecule is the substrate for the next round of oxidation, starting with acyl-CoA dehydrogenase. Repetition continues until all the carbons of the original fatty acyl-CoA are converted to acetyl-CoA. In the last round, a 4-carbon acyl-CoA is cleaved to 2-acetyl-CoA. Coming to the energetic of fatty acid oxidation, example for this palmitic acid, it's a 16-carbon compound. Beta oxidation of palmitic acid will be repeated 7 cycles, producing 8 molecules of acetyl-CoA. In each cycle, FADH2 and NADH plus H plus is produced and will be transported to the respiratory chain. So, 1 molecule of FADH2 will give 2 molecules of ATP, and 1 molecule of NADH plus H plus will give 3 molecules of ATP. It happens in 7 cycles, so 5 into 7 altogether 35 molecules of ATP. Each acetyl-CoA, which is oxidized in citric acid cycle, gives 12 molecule of ATP. So 8 cycle into 12 molecule of ATP, 12 is equal to 96 molecule of ATP. And 2 molecule of ATP are utilized in the activation of fatty acid, and it occurs once. So energy gain is equal to energy produced minus energy utilized. Energy produced is equal to 35 molecule of ATP plus 96 molecule of ATP minus energy utilized 2 molecule of ATP. Altogether 129 molecule of ATP is synthesized through beta oxidation of fatty acid. Thank you.